Nalco has recently launched a second-generation adenosine triphosphate method to enumerate bacteria in oil-filled samples. This is a Nalco-exclusive technology and is a differentiator that we can use in the field to get real-time feedback on microbial activity and risk in our systems. The advantage to the ATP kit, as opposed to serial dilution, is that this method is not based upon growing bacteria in culture, so there's no risk of missing organisms that simply don't grow in the culture models. Additionally, it takes approximately 5 minutes to run the test, and we immediately have that feedback and know the risk of the microbes in the system, as opposed to having to wait 14 or 28 days for the incubation to occur and the bacteria to grow and cause that color change. In this video, we'll walk through the steps for processing a fluid sample to enumerate bacteria using the second-generation ATP method. The first thing we do in this method is to calibrate the enzyme. This is very important because enzyme calibration will change depending on the day that the test is being run and how old the enzyme is. This occurs by taking 2 drops of the enzyme and mixing that with 2 drops of a calibration solution that allows us to detect how active the enzyme is. After 2 drops of the enzyme and 2 drops of the calibration solution have been mixed, they are put into the luminometer, and the luminometer allows us to read the light output of the activity of the enzyme. This is important as at some point in the life of the enzyme it becomes so inactive that it can no longer be used for the testing. So when the luminometer gives us a readout, which in this case it says the activity is 6,045, that lets us know whether or not the test can continue. A number below 2,000 is not considered acceptable for use and a new enzyme should be used. In this case, 6,045 has plenty of activity. At this point, we move on to actually processing our sample. So for this fluid sample, we're going to process 10 milliliters. Depending on your given system and depending on the microbial load, we often use somewhere between 10 milliliters and 50 milliliters of water from the system or fluid from the system to evaluate microbial activity. So that 10 milliliter sample is removed, a 0.2 micron glass silica filter is added, and we push that fluid through the 0.2 micron glass silica filter. That allows us to trap all the intact bacteria on the filter and allows us to push through all the ATP that is not inside of cells and is present just in the bulk fluid phase. At this point, we remove the filter, we take the plunger out of the syringe, we reattach the filter, and then we are going to add 5 milliliters of a solution that is used to remove inhibitors that are often seen in oil-filled systems. This can be the presence of oil, high chlorides, as well as different chemicals that are potentially present in your samples. That 5 milliliters is added, and that 5 milliliters of the inhibitor removal solution is pushed over the filter as well. This now allows us to get a reading that will be accurate and meaningful for the sample that we're testing. After that 5 milliliters is pushed through, the filter is again removed, and what we will now do is push 160 ml syringe worth of air over the filter. This simply allows us to dry the filter and remove any residual inhibitor removal solution that might still be present. At this point, we are ready to break open the bacteria and release the ATP that will be measured. In order to do this, we are going to reattach the filter to the 20 ml syringe, and we're going to add 1 milliliter of a detergent solution. That detergent, when passed over the filter, will break open the bacterial membranes, releasing the ATP that we will then measure in the subsequent downstream step. So we add 1 milliliter of the detergent solution, and we are going to push that milliliter through the filter into a new collection tube that will be used for our downstream measurement. What you will notice when you do this is that you will see some foam that comes out the other side. This is simply from the detergency that's added in the lysis solution. To that, we add 9 milliliters of ultralute. That is our dilution solution. In this case, it was pre-aliquoted into the tube, and it is mixed by inversion to ensure that it is homogeneous. Our sample is now ready to be tested to determine how much ATP is present in that sample that was processed. To do this, again, we will take 2 drops of the enzyme. The 2 drops of that enzyme are going to be mixed with a 100 microliter sample, or 0.1 mil, of our 10 milliliter sample that contains our free ATP that's been broken open and released from the detergent extraction. That 100 microliters is equivalent to the 2 drops of the enzyme. Those 2 are mixed together in the tube, and then that is again put into the handheld luminometer to determine how much ATP was present in that particular sample. The three most important pieces of information in this test are the activity of the enzyme that was calibrated up front, the volume of fluid that was pushed through the filter, and this final reading. In this case, the final reading is 59,494. Those three pieces of information are put into an Excel spreadsheet that Nalco provides with this kit, and that gives instant feedback on, number one, the microbial population in the system, but number two, also what that means in terms of risk for your system, whether it's a low risk, a moderate risk, or a high risk. This technology gives you that instant feedback that you need in your systems in order to be proactive, not reactive, to microbial risk that could be occurring in your system.